This is a report on a project that's in progress, or wasn't originally scoped out that way, but it's on denitrifying bioreactors, which Richard Cook is pioneering and advocating in this area. And it's addressing the question of whether they produce a significant amount of methylmercury, which would be a different kind of water quality problem than nitrate. Yeah, so anyway, I have to give acknowledgements since I have poor control over when I finish talking. So, first, of course, I want to thank ISCC for funding and for patience with the pace of our progress on this. It's actually worked out pretty well. We were supposed to be done a while ago, but because we stretched out, I've collected some more samples that will be analyzed, and we'll end up making the results a little more interesting. So what I am going to present today is a subset of the results that we've obtained so far, and even a smaller subset of the overall project once it's done. The analyses that I'll be showing were all done by Brian Vermillion, who left around a year ago, and since I lost my lab staff, I drastically slowed my pace of work. But anyway, he did all the methylmercury analysis, and then Richard Cook's group did some of the sampling and expertise related to the bioreactors. Okay, so this talk is really a tale of two elements. Both of them have complex biogeochemical cycles involving a range of chemical species. Nitrogen has gaseous species, aqueous species of different oxidation states, and solid species. Mercury has gaseous species, aqueous species of different covalent nature, or different molecular nature, and solid species as well. So two elements that have more complicated biogeochemical cycles than many pollutants do. Many pollutants might just have a couple of different species that are really important. They're also related or similar in that they both are non-point source pollutants. Nitrogen is a non-point source pollutant derived from fertilizer overuse. Mercury is a non-point source pollutant generated by coal combustion and waste incineration primarily. Of course, there are natural sources of both. The nitrate exported from agricultural fields impacts the Gulf of Mexico. It impacts water quality for communities that obtain drinking water. And of course, there are other ecosystem impacts that I will go into. Methylmercury, which is really the form of mercury that has the main impact that we're concerned about, strongly bioaccumulates in food webs and in aquatic food webs especially, and it ends up in fish. The consumption of fish is the main source of human exposure to mercury. Methylmercury is also the leading cause of freshwater fish consumption by adults in the U.S. And a significant percent of U.S. women of childbearing age have blood mercury higher than the U.S. EPA threshold level. Significant meaning 6 or 8% in a recent study. That doesn't mean it all came from freshwater fish, but a lot of it probably came from marine fish. But people are concerned about keeping freshwater fish levels low, both as a recreational resource and also just to avoid increasing levels of mercury in people in general, but women particularly, because the lowest level of impact is on the development of children and their neurological development. Okay. Another parallel between these two elements is that both have key processes in the environment that occur in anaerobic environments. One is, for nitrogen, it's denitrification, which alleviates some of the impact and consumes the pollutant that people are worried about. For mercury, it's methylation, and that is the transformation from mercuric ion to methylmercury. And this greatly increases the impact. So there's an opposite effect of these anaerobic processes on these two elements. I've shown here kind of how these processes occur in a stream. That's why I've got it slowed there. I know it's a little steep for Illinois, but anyway. Okay, so here we have the water up here, and we have mercury 2, and nitrate, and oxygen, and this here is a sediment, and down here would be groundwater. So the mercury 2 gets into sediments, and most of it's stored, but there's some dissolved mercury. And then, of course, nitrate, oxygen are diffusing out into sediments, and as the bacteria consume organic matter, they use up oxygen and nitrate, manganese, iron, sulfate, there's an order of carbons shown here. And because they get more energy from oxygen, of course. So these are all competing electron acceptors. And typically different bacterial populations carry out these reactions. Well, it turns out that mercury is transformed to methylmercury by sulfate reducers. That's the best known process, but iron reducers have recently been shown to also methylate mercury. Now there's a host of issues involved in this I'll get to in a second. But what's important about this is that because nitrate reducers compete better for the energy that's available in the form of organic compounds, they can out-compete iron reducers and sulfate reducers. Basically, nitrate inhibits sulfate reduction and iron reduction. So there's a competition between nitrate and nitrate actually ends up inhibiting mercury methylation, and this is documented in studies in the Adirondacks and lakes there, and undoubtedly affects Illinois as well. And I think it partly accounts for the fact that Illinois isn't known for having a big mercury problem. Okay. This hasn't been shown in the field. I think it's pretty basic, very strong case that can be made just from other field results and basic hydrogen chemistry, that our high nitrate levels end up decreasing the amount of methylmercury production. Okay, so that's just some of the background. One more thing I'd like to show is that, so there's really two issues in methylation. One is, does the organism have the right enzyme to produce methylmercury? We know for sure that iron reducers and sulfate reducers do. The other issue is how bioavailable is the mercury 2 that is the substrate for methylation. And I've shown here some of the basic chemical reactions that occur in oxic waters. This top reaction, you have organic matter, which I've depicted as RSH, with thiol groups binding mercury 2, which is primarily in mercuric hydroxide form in fresh waters, and will bind to form mercury bound to dissolve organic matter. And this is typically unavailable to microbes because it's a large molecule. If it's a large molecule, it's typically unavailable. There might be some small, like amino acid or something that would be available. So not every organic compound is unavailable. But large human compounds are typically unavailable. In the presence of sulfate-reducing bacteria, you get hydrogen sulfide form. And this is one of the strongest ligand that binds mercury. And basically, it will strip mercury out of the thiol complex to form this complex, which is bioavailable. Okay, so there's two real impacts of sulfate-reducing bacteria. The first is having the right chemistry, or the right enzyme. The second is altering the environmental chemistry to make mercury 2 bioavailable. Now, typically you would not think that this is bioavailable if you think in terms of free metal ions, because sulfide makes the free metal ion concentration really low. But it's because this is a passive species that can diffuse across membranes. Okay, I'm getting pretty deep into the theory here. But it's all relevant to issues. This is a the kind of data that shows how important anaerobic environments are for mercury. This is a profile that we obtained, had some guest students this summer, and I took them out to Clear Pond at Kickapoo State Park, and we obtained some water samples and measured the DO and temperature. We didn't get sulfide, but then we measured methylmercury. And I'm not sure if that sample is quite right, but it's close. Basically, we get 0.1 or less nanogram per liter up and down the water column until we get to very far into the anaerobic area. And then we get up to about 2 nanograms per liter. And that's very high. So the thing to remember here is nanogram per liter methylmercury is what you see in a very anaerobic environment. And 0.1 or so is what you see in an aerobic environment. Okay. This slide is from a recent paper on documenting a survey of river basins conducted by the US Geological Survey. And I don't want to go through all the data here, but note the axis here is wetland abundance, and then here's the methylmercury, filtered methylmercury, which is these triangular shapes. And you see across these basins, methylmercury is correlated with the percent of wetland area in the basin. So what am I saying? That it's these wetlands have anaerobic environments, right? So if the landscape is impacted by anaerobic environments, you get more methylmercury. It's just because upland environments don't export as much mercury, they don't generate methylmercury. Okay. So wetlands impact methylmercury at a basin scale. And that's the controlling factor of methylmercury in many cases. Okay. So, what does this mean for attempts to mitigate nitrate pollution by using constructed anaerobic ecosystems? This is a slide that shows a project out in Decatur with 2 different kinds of constructed anaerobic ecosystems. One is best known, which are these constructed wetlands. Okay, now, I'm not going to go into those, but that's a well-known type of system. And you definitely want to think about methylmercury if you're going to build a lot of constructed wetlands. The other one that's less well known, but I think is promising, is this one right here, which is a bioreactor, a denitrifying bioreactor, which Dr. Cook built and has installed in a number of places around the area. The denitrifying bioreactors are designed to reduce nitrate export from tile draining fields. They're economical, they have a small footprint, so you don't lose a lot of farmland. You put them right on the edge of a ditch, and that isn't prime farmland anyway. You fill it with wood chips, which are inexpensive. They have control structures, which are $5,000 to $10,000 to install. And it doesn't take a lot of time to operate and maintain. So it has some advantages over wetland. You don't give up as much area, and it's not that expensive.
Is this something I don't know?
Yes, I'll show you. And field tests show that they're highly efficient. So here's another bioreactor. This one is near Monticello. Here's I-72, and here's a ditch flowing here and a bioreactor right here. You can see the tile coming in off the field here, the bioreactor. Another tile coming in here. And this would be what the control structures look like at the surface. These are so it's a combination of steel and plastic. And at the top is steel. I believe the sides are plastic. I'm not sure what the plastic material is. Here's a cross-section of a bioreactor. So here's the control structure that you were just looking at. The tile comes in up here. Depending on how the control structure is set up, some of the water is diverted through here. And this is a cross-section of a big pit filled with wood chips. And then it comes out here. You have another, basically you have meters inside here that control where the water flows. So normally it's set up so that under low flow, the water is all routed through here. Under high flow, some of it is routed out here just so you don't overload the reactor. And then here you have a meter to keep the water level in the reactor at some desired level that flows out to the stream here. Here's where somebody photoshopped it into the side of the dish. Looks a little funny, but it maybe helps you visualize what's going on. And so you get a tile coming in and then flowing through the reactor and coming out. This nice little waterfall there. Okay, this is another sketch of the reactor, so you get a feeling. Basically, it's a big pit. Sorry, the dimensions aren't showing. 10 feet wide, and the length varies. It could be 100 feet, it could be 50 feet, and the depth here is 5 feet. And then in this particular one, this is the first generation design, or they call it second generation, but the first type that we sampled in the study. The water comes in, and then you can see that it would have to fill up to this level before it drains out. Okay? All right. This is a summary of the load reduction that Dr. Cook has documented for these reactors, for some of these reactors. And you can see that you can get 60, depending on how heavily your load, how much area is draining into a bioreactor and how big your bioreactor is. That's the loading density, acres per 100 square feet of a bioreactor. At high density, at a high loading, you get maybe 50% removal. At a low, when you have a lower loading, you can get 100% removal. So that gives you some idea of how these things perform. In other words, quite well if you don't overload it. So the theory of how this works is that you have the wood chips in there, the cellulose is broken down by fungi into small organic molecules, and there's bacteria, denitrifying bacteria that take the nitrate and use the nitrate as an oxidant to break down these organic molecules, and you give off nitrogen gas. Okay, so in our study, then, our goal was to determine whether denitrified bioreactors produce methylmercury due to the anoxic environments formed within the bioreactor. Our secondary objective was to look at total mercury. I'm not going to talk at all about that today. And we want to compare it to natural levels in the environment. Our study design was to collect samples when the reactors are flowing. They aren't flowing all the time. We sampled the inlet and the outlet simultaneously. We sampled periodically from summer of 2008 to June 2009. We sampled again in 2010. We preserved samples by filtration and acidification or freezing. And so we still have a lot of samples that we can analyze. Basically, we're going to select some of what we have and finish the analysis. We've analyzed a variety of solutes in many of these samples. And then we're analyzing solid mercury. And so we haven't come close to analyzing all of them today, mostly because we lost Brian and also because our system really is a bleeding edge system, not just leading edge, but bleeding edge, and we were bleeding pretty profusely for a while. So we're back online now. So here is indicating how we sampled the inlet. There's an issue of sampling. How cleanly can you sample? That's a big issue in mercury analysis. For methylmercury, contamination is not such a big problem. It's more for total mercury is an issue. And in many cases, when you sample, we have to lower a bottle down into where the water is. So we're lowering it, 5 or 6 feet down into this well. And it's pretty hard to, it would be pretty hard to do and be absolutely clean for total mercury in those kind of sampling. So where possible, we sample from the outlet tile because we can just catch the water flowing out from this tile. Similarly here, we sample in the outlet tile where possible, but sometimes we have to sample in the inlet structure. Okay, so I have to do how am I doing on time? Okay. A quick survey of the method that we're using. The method for methylmercury analysis was developed by Chris Shea, myself, Chris did the lion's share of the work on that, and that's published in 2005 in the S&T. We have a patent for bright vermilion, and I developed a sample preparation for water samples, which is key to what we're talking about here. The key chemistry in this process involves phenol-thione switch, which is a pH-sensitive switch that shifts the relative preference for solutions containing thiourea and thiol resins for mercury and for methylmercury. So at high pH, mercury absorbs the resin. At low pH, it goes back into solution with thiourea, and so we can switch it back and forth between solids and solution phase that way. And that's a key part of this, a key step in this process. The second key thing is that thiourea complexes in the aqueous phase of methylmercury and mercury 2+ have a different charge, since thiourea is a neutral species. And so we can separate using an ion chromatography column. So that's the basis of the speciation of the chromatographic step. So here's a sketch of the system. I'm not going to go into the details, except to say that it's pretty complicated. And lots of things can go wrong to look at it. But Brian succeeded in generating a lot of really excellent data. That's Brian right there. And our method has been published, for sample preparation, has been published in Analytical Biochemistry. If we compare, Chris Shea also developed a method of preparing samples from Biota. We've worked out sediment methods, so we can basically handle all the key environmental samples. And if you compare the certified value of methylmercury in Biota and sediments to what's measured in what we measured, you see that the relationship is really good. We get the same thing essentially for these types of samples. I want to quickly go over. So basically, when you're handling a sample, you have to do ultra-clean sample collection, sample preservation. You have to extract the methylmercury from the matrix. You have to pre-concentrate the methylmercury. You have to chromatograph to separate and then to detect it. And if you look at the steps in the process for the standard method for water versus what we're doing, the standard method for water is distillation. We use thiourea catalyzed solid phase extraction. Then they do ethylation, purge and trap. We have some simple room temperature steps. This is distillation is a 100° centigrade step. This is room temperature. They have gas chromatography. We use ion chromatography, and then we have the same detector at the end. We've done some comparisons with Trent University, which is a very highly regarded lab. So this is the distillation ethylation value. Here's our value down here. You see that at high levels, these are from like the bottom waters of lakes, we get extremely close to the same thing. In this mid-range, we tend to get somewhat higher numbers than what they're getting with distillation ethylation, and we've been looking into why that is, and I'm going to just, for now, just say we're right. This is a more comparison with the USGS Wisconsin District Mercury Lab, and again, you see the same, similar thing. Well, it's not exactly the same, but we're getting some differences again. They're using standard method distillation ethylation, but we're not grossly different. Okay, so the bioreactor results. These are the inlet samples from the summer of 2008. Basically, nothing or very little in the inlet. 8 non-detects, maximum of 0.16 nanogram per liter, average of 0.09. That's really what you expect for groundwater. You don't expect to see anything in groundwater, which is what the inlet to the bioreactor should be giving. The outlets for the same bioreactors. This is Monticello. This is the Decatur site. And you see that we range from 2 to 10 or 12 nanogram per liter. In other words, these bioreactors were putting out methylmercury at a level that's typical or high even for an anaerobic ecosystem. okay? That's just to look at a little more detail. This is the bioreactor. I'll show you the data for next. This is the nitrate and flow data for the year of 2008. Here's the spring and winter and spring. You see that nitrate is quite high in the 5 to 9 milligram per liter range. And coming out of the bioreactor, it's knocked down a little bit, but not completely. Then by June, or by late June, nitrate disappears from the outlet. And so here's the efficiency. You can see the efficiency is around 40 to 70% or so until here. Now, it's not purely a function of flow, right? Because the flow is still reasonable in here, higher than the this period. So it's not purely flow, it's got to be temperature. And I can tell you just from looking at samples, when you look at the water coming out in the summertime, it's brown with, looks like beer, a nice dark beer. And in the wintertime, it's clear. So there's a lot of organic matter coming out. And the chemistry is really changed inside. Here's methylmercury from that same reactor. Here's the nitrate, the same nitrate data. Here's the methylmercury data. The green are the inlets, red are the outlets. And you see that the methylmercury is quite high. Again, characteristic of an anaerobic ecosystem. So, what's going on? Well, in groundwater, total mercury is very low. Typically, half a nanogram per liter or less. Methylmercury is very low, often undetectable. Surface waters, total mercury is 1 to 5 nanogram per liter. Methylmercury is less than 0.5 in unpolluted systems. So we're getting an elevation, and then I showed you that profile from the lake where it's 1.5 or so in bottom waters, and we look at other lakes, we get 2 nanogram per liter at the very bottom of a stratified lake where you have sulfate reduction. And clearly the bioreactor is behaving like a sulfate or a highly reducing system. This is too few data to be too excited about, but I thought I'd show it. There is something of a relationship between the increase in dissolved methylmercury across the reactor versus the decrease in sulfate. So perhaps it's because of sulfate reduction in those bioreactors. Even though they're not designed for sulfate reduction, you're going to have some places within the reactor that get more reducing than others, and so you clearly get some sulfate reduction in there. Whether it's the sulfate reducers or not, that's carrying out the reaction or not, I don't know. The other thing that's happening is the VOC is increasing a lot, factor 10 essentially across the reactors where we check VOC. And that's organic matter is often a carrier for mercury, especially when the organic matter has flyovers. Now, I'm not ready to say that the VOC coming out of these bioreactors is comparable to human material over this time. But it certainly is correlated with increases in VOC. Okay. So we could have some production, some combination of increase in VOC and decrease in sulfate may be responsible. And the levels in the bioreactors are high. How is this happening? What's the source of mercury? Groundwater is typically very low, except when you get preferential flow. So one scenario is that under preferential flow, that is high flow in the spring, the mercury is being carried into the bioreactor, absorbs on the wood, and then gets released in the summertime when it gets hot and the chemistry is right. Now, that's one scenario. The other source could be the wood chips. And we haven't, that is, trees accumulate mercury from the air and soil water into the wood, and that wood, as the wood chips break down, they could be releasing mercury too. So that's something obviously we need to check. I'm going to skip this one now, so I'm running out of time. So there's obviously lots of questions. What controls the extent of methylation? can the reactors be designed to minimize mercury formation? Richard has already implemented a change in design. Here was the original design that I showed you. You see that there was a pool of water that would sit down here all summer. And so what he did was he basically eliminated that pool so that the water shouldn't sit here all summer. And so we now have samples collected from reactors that have been set up to be to operate this way. And I can't give you this. I don't have a lot of results yet, so I'm not ready to talk about it. So, methylmercury is produced in some bioreactors. The fact that mercury fluxes in watersheds needs to be considered. I think as an analogy to that graph I showed earlier, if you have a lot of wetlands, you're going to get methylmercury in the streams and lakes, and the same thing would happen if you have a lot of bioreactors to consume nitrate. You could make the waters downstream. It could impact mercury levels in the ecosystem downstream. Can we reduce that methylmercury production? I think I'm sure that there can be some optimization there. Eliminate the ponding zones, like Richard already did. Might need to switch to low mercury wood. And that's it for now. Thanks again.
Yeah. Did you mention at the start of your talk that nitrate inhibits methylation?
Yes.
So
why? Yeah. See, that to me, that's partly why it's a surprise that this is happening. And so there's two explanations I can think of. One is that there are zones within these bioreactors where the nitrate is completely consumed, and so then in that zone you start getting iron reduction, sulfate reduction, and methylation. Okay, so that's one scenario. The other scenario is that, remember the slide about bioavailability that I showed, and how the hydrogen sulfide makes the mercury bioavailable? Okay. What really hasn't been looked at much is how widespread the enzyme. People don't even know what enzyme is doing the methylation yet. So we can't say, is it something that we expect to be widespread among bacteria or not? Okay. Now, if it is widespread, then the reason sulfate reducers seem to control methylation may be primarily their production of hydrogen sulfide and making it bioavailable. Okay, and so then, and because iron reducers are very closely in close proximity, live in close proximity, they may end up producing methylmercury also. So how does that apply to the bioreactor? Well, the organic matter in the wood has not gone through, as it decomposes, has not gone through all the same processes as human material in soils and in aquatic ecosystems. So it may not have the same ability to make mercury bioavailable. So in other words, you may not need as much sulfide production to make the mercury bioavailable. Okay, so it may not be as dependent on sulfate reducers as it is in natural systems. So that would be the other way it could happen.
Can you use plastic beads or balls instead of wood chips in the bioreactor?
You need the wood provides the organic carbon for the bacteria to grow.
So that is the, okay, I see.
Yeah.
So, but have you done a mercury analysis on the wood itself? That should be a simple thing.
No, I've been trying to get everything else done, but okay. I mean, you know, there's literature values on wood, and it's plausible that that's a source.
I think